Right, so we're gonna be looking at the respiration, right? So respiration and ATP synthesis, right? So it's generally the backways or the backward process for the reverse reaction rather for photosynthesis, right? So let's have a look at this. So we know cellular respiration in and of itself, right, to be the processes within generally living things, right, to convert glucose into energy. So autotrophs would produce glucose, right, or different types of sugars during photosynthesis, and then our heterotrophs themselves will actually break down these large molecules, right, into simpler molecules, right? So while the process seems complex, right, we're going to be looking at each part of cellular respiration. We know that cellular respiration occurs within, does it occur within the mitochondria? Does it occur within the cell, the cytosol? Right, we're looking at the mitochondria for the most part, right? The mitochondria is really the main area, right, for cellular respiration, and we need to know the structure of of a mitochondrion and understand the structures within a mitochondrion, right? So we'd have known that it is a double membrane protein as well. Well, not protein, organelle as well, where it has its different spaces. So it has its outer membrane, its inner membrane, right? And then you have an outer area, right? So that's outer space there, right? Within the inner membrane itself, the inner membrane boundary, right? It has a lot of folds in it, which we call cristae, right? And within the matrix, it is actually filled with mitochondrial DNA, ribosomes, different types of matrix granules, etc. Right? So we have that there. For the main process, right, of the electron transport chain, it occurs on the membrane, right, the inner membrane there of the mitochondria, right? So can anybody just let me know why the cristae are shaped like that? The cristae being folded, what is that primarily for? Go ahead. So it increases the surface area, right? So within every mitochondrion, right, the cristae would be folded like that in order to increase the amount of intermembrane, the intermembrane boundary, in order to increase the area in which you can have all these different types of peripheral and integral proteins, right, along the membrane. So increase the number of sites that respiration can occur. Okay, so generally looking at that. So for aerobic respiration, the release of energy during cellular respiration, we're looking at this, right? This is a general thing that we've been looking at, right? So we're taking our sugars now and using oxygen in order to oxidize them, right? And now produce carbon dioxide and water, right? But we know that there are four general steps here. So we have glycolysis, right, in which we have the photophosphorylation and splitting of glucose. Well, not photo, rather phosphorylation, my bad, right, and splitting of glucose. This occurs within cell cytoplasm, right? Within the matrix of the mitochondrion now, we're going to have the link reaction. We have the decarboxylation, dehydration of pyruvate, right? Then again within our matrix of the mitochondria, right, we're gonna have the Krebs cycle or the tricarboxylic acid cycle, the cyclic pathway with enzymes that control these reactions, right? And then within the inner membrane of the mitochondrion, we're gonna have our oxidative phosphorylation, the production of ATP through oxidation of hydrogen atoms, which is a form of chemiosmosis as well. All right. So before I move on, stage 1, stage 2, stage 3, stage 4, could you guys just let me know which stage causes the most issues? So which stage is a major problem? So type the number of the stage in the chat to let me know which stage I need to focus on a little bit more, and which stage I can not explain, but like expect questions for. So what are the stages that give the problems here? Is it glycolysis, link reaction, Krebs cycle, or oxidative phosphorylation? Alright, I'm not seeing anything. Alright, so everybody's good with this topic then? Okay, so Wanda said the link reaction. Tony said the link reaction. Well, not the link reaction. Krebs cycle. Both of you guys said. So the Krebs cycle is the main issue. So no one has any issues with glycolysis or anything like that. Okay. All right, interesting. So let's just start to review this. So we'll review the glycolysis and link reaction, and then focus on the Krebs cycle, right? So glycolysis specifically, right? We're looking at the process from glucose to pyruvate, right? So we know that glycolysis is one of these metabolic processes that really occur, right, in both aerobic and anaerobic respiration, right? So we're looking at the conversion of glucose to pyruvate, right? A six-membered ring molecule, right? We're changing it up, right? So the glucose is a six-membered ring molecule, right, found within the blood, and it usually results from the breakdown of carbohydrates into sugars, right? So it's usually the result of that breakdown, right? So after digestion, you're gonna have all of this glucose entering into the body, right? With the use of insulin, releasing it, well, not insulin, glucagon, taking it into the body, right? Into the cells specifically, right? And within the cell cytosol, we're gonna have glycolytic enzymes that are found within the cytosol, and we're gonna have this entire reaction occurring. So the overall reaction for glycolysis is provided down here, right? So we're looking at this. Alright, so we have a glucose and 2 NAD, 2 NADP, and 2 inorganic phosphates. Or we're going to end up with these 2 pyruvic acids, right? Some of these substituents as well. Alright, then we're going to have the 2 ATP, 2 NADH, and the excess protons. So we're going to be looking at this. So this is really a guide to really go over, but we're gonna be looking at generally here. All right, we're gonna start with here, going through all the steps specifically. All right, so step 1 at hexokinase. All right, the first step of glycolysis would be the conversion of D-glucose, right? That's the one that our body can use, right? Into glucose 6-phosphate. So this enzyme, this interaction is catalyzed by hexokinase, of course, right? So here the glucose ring is going to be phosphorylated, right? So you can see the process here. It's going to be phosphorylated via phosphorylation, right? Which is adding the phosphate group to the molecule, and that phosphate is coming from an ATP. So here glucose, right, is going to react with the ATP. It's going to release energy using the phosphor to actually bind, right, to the molecule here. So it's replacing the hydrogen on the hydroxyl group of the 6th carbon, right? Replacing that, we're gonna have a displacement reaction, creating our glucose 6-phosphate, right, and our ADP plus our proton, right? So that's it there. So the P is actually the phosphate group, replacing it with the phosphate group. All right. So our entire phosphate group goes there. The step 2, which actually here, right, is going to be catalyzed by phosphoglucose isomerase, right? The second step is going to be our glucose 6-phosphate, or G6P, turning into our fructose 6-phosphate. So we know that fructose and glucose are isomers from unit 1 already. Done and dusted, right? So what we're going to have is an isomerization, right? So we're going to have isomerism occurring, in which the phosphoglucose isomerase is going to be changing some of the glucose 6-phosphate into fructose 6-phosphate, right? So that's really the second step here. Okay. Step 3 with phosphofructokinase, we're gonna take now, right, our fructose. We're gonna be breaking that, right? So with a magnesium as the cofactor for this enzyme, right, it changes fructose 6-phosphate into fructose 1,6-bisphosphate, right? So it changes it now, fructose 6-phosphate to fructose 1,6-bisphosphate, right? And this occurs also with ATP. So once it had only one phosphate group, now it has 2 phosphate groups, right? Creating fructose 1,6-bisphosphate. Alright? Then step 4, catalyzed by aldolase, right? We're going to have the splitting. Aldolase is a kinase enzyme. Well, it's a chelating, not a chelating enzyme, a kinase enzyme, right? So it cuts, right? It's a lysing enzyme. So what it's going to do, it's going to split fructose 1,6-bisphosphate into 2 different sugars. We're going to have dihydroxyacetone phosphate, and we're going to have glycerol 3 phosphate, right? So we're gonna split it now. So we're splitting the fructose 1,6 bisphosphate into our dihydroxyacetone phosphate and our glyceraldehyde 3-phosphate, right? And then we're gonna have this reaction. Step 5, triphosphate isomerase, right? So both are triphosphates. What we're gonna do is try to change our dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, and that's what the triphosphate isomerase does. It isomerizes the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. That's what's happening, right? So here now we have 2 glyceraldehyde 3-phosphates, and notice in this reaction now we have actually the consumption of ATP. This is the energy, this is not the payoff phase, this is the energy investment phase, right? So first we have hexokinase, right, actually replacing a phosphate group with the hydrogen on the 6th carbon of glucose, creating 6 glucose phosphate, or what we call glucose 6-phosphate, right? Then we're gonna have phosphoglucose isomerase changing the glucose 6-phosphate into fructose 6-phosphate. Then we're gonna have a phosphofructokinase changing our fructose 6-phosphate into fructose 1,6-bisphosphate, and then we're gonna end up with aldolase, right? Actually breaking the fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then triose phosphate isomerase is gonna actually change our dihydroxyacetone phosphate into our glyceraldehyde 3-phosphate, and now we end up with 2 glyceraldehyde 3-phosphate molecules. All right, so that is what the energy investment phase is. We're using ATP to change the glucose, to change the glucose, so fructose 1,6-bisphosphate, then break it, right, and then make sure that we have some amount of configuring, some shifting of some isomerism in order to make sure that we end up with glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is what we're going to be using in the energy payoff phase, right? So the investment phase is all about rearranging molecules, right, in order to create 2 glyceraldehyde 3-phosphate molecules, all right? Within photosynthesis, is actually the backwards of all of these reactions will be occurring in order to create glucose, right? So for glycolysis now, we break down the glucose back to the G3P, right? It's the same thing that we saw in the Calvin cycle. G3P that is left behind to create glucose would have gone through these processes to create glucose. What we're doing is reversing them in glycolysis, all right? Now we end up with the payoff phase. What we're looking at now is the production of NADH and the production of ATP, as well as the pyruvate within these last steps, right? So now we're looking at the path of 1 glyceraldehyde 3-phosphates, right? That's what we're looking at now. So all these steps occur twice, from step 6 to step 10. To steps 10, right? So 6 to 10 occurs twice. Please remember that. What we're looking at is the path of 1 of the glyceraldehyde 3-phosphates. Just remember that this happens twice for the 2 glyceraldehyde 3-phosphates, right? So step 6, we have glyceraldehyde 3-phosphate dehydrogenase, right? And what does this do, right? So dehydrogenase would dehydrogenate, right, our G3P, and adds an inorganic phosphate to the glyceraldehyde 3-phosphate, producing 1,3-bisphosphoglycerate, right? So with glyceraldehyde 3-phosphate dehydrogenase, right, we change G3P, right, into 1,3-bisphosphoglycerate. That should be fine. And we have the production of NADH here. Alright, so we have the production of NADH within this step. For step 7, we have phosphoglycerate kinase, right? So in this case, now the kinase transfers the phosphate group from 1,3-bisphosphoglycerate, right, to ADP to form ATP, right? So 3-phosphoglycerate. So we end up now with 3-phosphoglycerate, and we have the production of 1 ATP molecule. Alright? Notice that these occur twice, so technically we already have 2 NADHs and 2 ATP molecules. Right? For step 8, phosphoglycerate mutase now will mutate the molecule, it will change the molecule. What it does is essentially changes the position of the phosphate group from the 3rd carbon to the 2nd carbon, now changing 3-phosphoglycerate to 2-phosphoglycerate. That's all it does. Really, really simple reaction, right? We just change which carbon the phosphate group is on. All right. Now we're gonna look at enolase, step 9 with enolase. This actually has the creation of water, right? So the enzyme enolase removes a molecule of water from the 2-phosphoglycerate to form phosphoenol pyruvic acid, right, or phosphoenol pyruvate, right? So remember, if it's hydrated, it's going to be pyruvic acid. If it's not hydrated, it's going to be pyruvate. The difference between the acids and the ates is just that one is an acid and one is a salt. So pyruvate is a salt, pyruvic acid is the hydrated salt because it's an acid salt. All right. So details basically in this step involves the conversion of the 2-phosphoglycerate, right, to phosphoenol pyruvate, right. The reaction is catalyzed by the enzyme enolase, and enolase works by removing a water group, right. So that's what the enolase does, right. It takes water from a molecule. So when we look at enolase, we just remove the water from 2-phosphoglycerate. All right. So here now we have phosphoenol pyruvate, and then at step 10 we have pyruvate kinase. So this is the last step of glycolysis, where we have phosphoenol pyruvate changing into pyruvate, right, with the help of the enzyme pyruvate kinase, right. As the enzyme name suggests, the reaction involves the transfer of a phosphate group, right, so in the kinase family here. So the phosphate group attaches second carbon, right, of the phosphoenol pyruvate is now transferred to a molecule of ADP, right, producing yet again another molecule of ATP, right? So we have that there. So that's really the entire process, right? We start off with hexokinase, changing glucose to glucose 6-phosphate. Then we have the isomerases, right? Changing glucose 6-phosphate to fructose 6-phosphate. Then we have a fructokinase, right, which changes fructose 6-phosphate to fructose 1,6-bisphosphate. And an aldolase will now break it into two, giving us G3P and dihydroxyacetone phosphate. The dihydroxyacetone phosphate is changed via triose phosphate isomerase into G3P. Then these steps occur twice. So glyceraldehyde 3-phosphate will now change into 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. Then we're gonna have phosphoglycerate kinase actually taking one of the phosphate groups, right? That will be from the first carbon, right, and creating ATP, leaving just 3-phosphoglycerate, right? That 3-phosphoglycerate now will have the phosphate group moving from the third carbon and placing it on carbon number 2, changing it from 3-phosphoglycerate to 2-phosphoglycerate. Then we have enolase removing our water, creating our phosphoenol pyruvate, and then pyruvate kinase removes the last phosphate group, which will now produce another ATP molecule, and finally pyruvate, right? So that is the entire process there. Now to have a look at the fate of pyruvate specifically, right? What happens to pyruvate after this? Could anybody let me know? What happens to pyruvate after glycolysis?
Sir, it enters the Krebs cycle.
Okay, it can enter the Krebs cycle. Any other takers? What happens to pyruvate after glycolysis? So it enters the Krebs cycle, okay. In what case? In what case would it actually enter the Krebs cycle? Well, through the link reaction, lovely Laquanda, we'll have to go through that first. But go ahead. Somebody unmuted, I believe. Under what conditions will it enter the Krebs cycle? Will it go through the link reactions to enter the Krebs cycle? Under what conditions?
Aerobic.
Aerobic conditions, lovely. If it's not aerobic and it somehow is within an, let's say, an amoeba or within a human, what will it go through?
Fermentation.
What type of fermentation?
Anaerobic fermentation?
Well, all fermentation are
Lactose?
anaerobic. Lactic acid fermentation, lovely. Right? Within other types of prokaryotes, right? And within some, well, most notably plants, we're gonna have ethanolic fermentation or alcoholic fermentation, right? Lovely. So after glycolysis, we're gonna be looking at that, right? The cell must continue respiration, either in aerobic or anaerobic conditions, right? The choice is based on the circumstances of the particular cell, right? So a cell that can perform aerobic respiration and which finds itself in the presence of oxygen will continue through the citric acid cycle, right, or the Krebs cycle in the mitochondria, right? If a cell is able to perform aerobic respiration in the situation when there is no oxygen, in the case of muscles under extreme exertion, we're going to have anaerobic respiration, right, called homolactic fermentation, right? So some cells such as yeast are unable to carry out aerobic respiration, therefore they will automatically move to anaerobic respiration called alcoholic fermentation, right? So within humans, we have homolactic fermentation, what we call lactic acid fermentation. Within yeast and other types of plants, etc. Right? In plants, I think this lactic acid fermentation, no, not lactic acid fermentation, alcoholic fermentation will occur regardless, right? So, but we're primarily speaking about yeasts, right? Organisms like those, right? Those type of cells, as well as large heterotrophs like humans, right? So it's either we have lactic acid fermentation or alcoholic fermentation, or in the presence of oxygen. If the cell can actually go through the Krebs cycle, then it will go through the Krebs cycle, right? So that's what happens after glycolysis.